Okay, everybody, close your eyes. Do that and tell me, what do you see? Do you see nothing? Complete darkness? Complete blackness? If that is the case, my question to you is why? Why do you see nothing when you close your eyes, or why do you see nothing in darkness? For us to understand that, we have to understand what happens during the darkness and what happens when the light rays enters into your eyes. I think this is a no surprise that all of you know that your eye helps in vision, right? Light rays from the outside world enters through the eyeball till it finally reaches the retina, where you have the photoreceptor cells that helps in the perception of light. Just like any stimulus on your skin helps you to perceive the sensation of touch or pain, similarly the stimulus of light helps the retina for the sensation of vision, right? So let's discuss some general anatomy of the eyeball. Eyeball consists of three main layers. The first layer is the outermost layer is the fibrous layer, which in the posterior 5/6 part, that is opaque, known as sclera, and in the anterior 1/6, this is transparent, known as cornea. The middle layer is the choroid layer, which is the vascular layer. This contains the blood vessels, and the innermost layer is the retina. This innermost layer contains the receptors for the light, for the stimulus of light. They are called photoreceptors. They are present on the retina. But that's not that simple. When light passes through the retina, all of this is retina. If you look at here, the innermost layer of the eyeball, this retina, this consists of 10 layers, and when the light from the outside passes through the pupil, lens, aqueous humor, and vitreous humor till it finally reaches the retina, before it can actually reach the photoreceptor cells that are rods and cones, it has to pass through these different layers: ganglion cell, plexiform layer, nuclear layer, so that it can finally reach the photoreceptor, that is rods and cones. These are the receptors for the stimulus of light, and they generate electrical impulses from the light energy, right? Because at the end of the day, electricity is the language that your brain understands. Everything has to be converted into electrical impulses for your brain to perceive, right? The sound that you are listening right now, your ear collects the sound and converts it into electrical impulses and then sends it to the brain. Everything that your brain perceives, interprets is electricity, because electricity is the only language that your brain understands. Now, light passes through all these layers before it can actually reach the photoreceptors, right? And then on the outermost part of the retina, we have the pigmented layer. This consists of melanin. This makes the pigmented layer black. Why this is black? The main reason is because when light rays enters, there is a chance that it might get reflected within the eyeball and reach another part of the eyeball, which can interfere with our visual acuity, right? So this pigmented layer, when the pigmented layer is black, this helps to absorb the light. This prevents the reflection and helps to maintain the visual acuity. In people with albinism that lacks melanin, this pigmented layer is not that black, and for that reason, the light rays that strikes into the retina gets reflected within the eyeball, and they have problem with the vision. But our main concern right now is with this layer, the layer of photoreceptors, rods and cones. Rods actually helps us in the scotopic vision, that is in the dark vision, and cones helps in the colorful vision, bright vision, right? And these receptors for the light, rods and cones, this consist of visual pigments. Rods consist of rhodopsin, whereas cones consist of different pigments which are not fully understood, but they are called colored pigment or the cones pigment. If you look at any one photoreceptor on a detailed view, then this consists of outer segment, inner segment, nucleus, and a synaptic body. This synaptic body synapses with the bipolar cell. If you look here, this synaptic body either synapses with the bipolar cell or with the horizontal cell. So again, the fourth part: outer segment, inner segment, nucleus, and the synaptic body. Outer segment consists of visual pigment, that is rhodopsin in rods and color pigment in cones. We know very little about the visual pigment of the cones, but rhodopsin is well understood, and we'll be discussing in terms of rhodopsin. But the same mechanism follows for the pigment in the cones as well. So we want to discuss what light does to the photoreceptors, and particularly to these visual pigments, rhodopsin. And now, the inner segment of the rod consists of cytoplasm and the cytoplasmic organelles. The important one is mitochondria, and then we have the nucleus and the synaptic body, right? Now, rhodopsin is a chromoprotein. Rhodopsin was present in the outer segment of the rod, right? That is a chromoprotein that consists of two portions, chromophore and a protein. The chromophore portion of the rhodopsin is 11-cis retinal. This portion of the rhodopsin is colorful, it absorbs light, and then get activated into many different intermediate products until it finally gets converted into transretinal. But the main problem with the transretinal is that protein component of the rhodopsin, that is opsin, can remain attached only with the cis retinal. When the rhodopsin gets activated and converted into transretinal, this opsin cannot stay attached to the transretinal because of the changes in the chemical configuration. So that's why transretinal and opsin get dissociated. And the transretinal is not colorful, it is colorless. And since the rhodopsin loses its color, this process is also called bleaching. So this is what actually happens. We have visual pigment rhodopsin in the outer segment of the rod, which is nothing but the combination of 11 cis retinal and opsin. This is a chromophore portion and this is a protein portion, right? Opsin is also called scotopsin because the rods help in scotopic vision or the dark vision, right? Opsin and 11 cis retinal combines to form rhodopsin, which is the visual pigment. And when light reaches the rods, this rhodopsin gets activated into many different intermediate products within the fraction of seconds, like vathrhodopsin, lumirhodopsin, metarhodopsin first, and metarhodopsin second. This metarhodopsin second is the activated rhodopsin that generates electrical impulses that is carried upon by the optic nerve to the brain, right? Now, after metarhodopsin, it gets converted into 11 transretinal and opsin. But opsin cannot remain attached to the trans form of the retinal. It can only stay attached with the cis form. That's why transretinal gets separated and opsin gets separated after the formation of metarhodopsin, right? But at any point, we require this visual pigment, rhodopsin, for us to see, right? So this transretinal somehow has to be converted into cisretinal so that the rhodopsin is regenerated. For this purpose, we have the enzyme retinal isomerase. This retinal isomerase converts trans form of the retinal into the cis form, and now the cis retinal and the scotopsin combines again and forms rhodopsin until they are again bleached by the light again. And if that happens, when light strikes again, this overall process continues again and the rhodopsin keeps generating again and again. And this process is also called the Walsh visual cycle because the degeneration and the regeneration of the rhodopsin keeps going. There is another process by which the rhodopsin can be regenerated, that is through the conversion of transretinal into transretinol, which is a form of vitamin A, and this again gets activated by the enzyme isomerase to gets converted into cisretinol and cisretinol, and that's how rhodopsin get activated by the light, converts into transform. Transform cannot bind with the opsin, so cis form has to be regenerated. The enzyme retinal isomerase helps in the regeneration of cis form that combines with the opsin and generates the rhodopsin. So this keeps going. But to understand what happens during the light and during the night, we have to understand what happens when this rhodopsin gets activated. To understand that, we have to look at the outer and the inner segment of the rod. In the outer segment of the rod, we have a sodium channel on the outer segment of the rod that is cyclic GMP gated. This sodium channel is kept open by the cyclic GMP, and when there is decrease in the cyclic GMP, this sodium channel gets closed. Now, in the inner segment of the rod, we have sodium potassium ATPase. This sodium potassium ATPase is continuously pushing the sodium out of the rod and bringing the potassium ions into the rod. But on the other side of the inner segment of the rod, we have a potassium channel that is continuously pushing this potassium out of the rod, right? So we are losing sodium as well as potassium in the inner segment of the rod. So the inner segment of the rod is always negative. But in the outer segment of the rod, when there is enough cyclic GMP, this sodium channel is kept open. When there is decrease in the cyclic GMP, the sodium channel remains closed. When the sodium channel is open due to the enough presence of cyclic GMP, sodium ion enters into the outer segment of the rod, and this creates some kind of positive potentials on the outer segment of the rod. But the inner segment of the rod is always negative because sodium is continuously being pushed out by the sodium potassium ATPase, and the potassium that is entering into the inner rod segment is also being pumped out through this potassium channel, right? So inner segment is always negative. When sodium ion enter through the cyclic GMP gated channel, this negative potential decreases, it becomes less electronegative. But when this sodium channel remains closed, we do not have any influx of sodium. We are only losing sodium as well as potassium. We are losing the positive ions, so the membrane potential becomes hyperpolarized. Hyperpolarization occurs when this sodium channel, cyclic GMP gated sodium channel, is closed. Now the main question is, when does this cyclic GMP gated channel, cyclic GMP gated sodium channel remains open, or when does this remain closed? So to understand that, we have to understand this picture. We are magnifying on one of the outer segment molecule. When the light strikes, we already discussed that the rhodopsin gets activated. We talked about all the change in the configuration of cis-retinal into transretinal, and then the regeneration of the cis-retinal by the retinal isomerase. So light activates rhodopsin, and when rhodopsin gets activated, it activates transduction, that is in the outer segment of the rods, right? Transduction is nothing but one of the G protein, a trimeric G protein that consists of 3 subunit, alpha, beta and gamma. It's a trimeric G protein, and like many other G protein, this transduction, a G protein, activates phosphodiesterase. Phosphodiesterase hydrolyzes cyclic GMP and converts it into 5' GMP. That means when phosphodiesterase is activated, we are losing cyclic GMP. It is being destroyed or it is being converted into 5' GMP. And when the cyclic GMP is being decreased, the sodium channel was cyclic GMP gated, and this was kept open by cyclic GMP. Since we are losing cyclic GMP because of the presence of the light, this cyclic GMP gated sodium channel remains closed now because cyclic GMP was helping to keep this open. And now when this sodium channel, again, when this sodium channel remains closed due to the light, the membrane potential, the overall membrane potential of the rod is hyperpolarized, right? Because in the inner segment we are continuously losing sodium as well as the potassium, but no amount of sodium is entering into the outer segment of the rod because of the decrease of cyclic GMP due to the presence of light, right? But during night the rhodopsin, during the absence of the light, rhodopsin is not activated, so that means G protein is not activated, that means phosphodiesterase is not activated, that means cyclic GMP is not destroyed. So there is increase in cyclic GMP during the night. So this cyclic GMP gated sodium channel is kept open, sodium ion can enter here. It reduces the overall electronegativity of the rod's membrane potential. So rod become partially polarized during night. It is hyperpolarized during the presence of light, right? Again, the same thing. During the dark, we have high amount of cyclic GMP because in the absence of light, rhodopsin is not activated, so cyclic GMP is not hydrolyzed. We have high cyclic GMP and the sodium channel opens, sodium enters into the outer segment of the rod, and it decreases the overall electronegative potential. During the light, cyclic GMP is low, so due to the low cyclic GMP, this cyclic GMP gated sodium channel remain closed, sodium cannot enter, and because in the inner segment we are continuously losing sodium as well as potassium, the overall membrane potential of the rods during the presence of light is hyperpolarizing. And this is a very interesting concept because in any other receptors of the body, it is the depolarization that conducts nerve impulse, but in the rods or the photoreceptors, rods, cones, it is hyperpolarizing membrane potential that conducts the nerve impulse. Everything that we discussed is just mentioned in words here. If you want to read, you can read it by yourself. The thing is, during the dark, cyclic GMP increases, sodium channel opens, and the overall electronegativity during the presence of light, if it was up to minus 70 millivolt, this electronegativity decreases to minus 40 millivolt due to the influx of sodium, right? So the overall electronegativity decreases during dark. That means this is partly polarized. During the presence of light, it is hyperpolarized. During the absence of light, it is partly polarized, and this partial polarization causes the release of neurotransmitter by the rod. What I mean to say is, if you look at this structure of the rod, from the synaptic body, the rods release the neurotransmitter because of partial polarization, because neurotransmitter is released during the polarization, right? When this becomes less electronegative, or when it becomes more positive, more amount of neurotransmitters are released. So rods during the dark, during the absence of light, secretes neurotransmitters. And if you look at this picture, the secreted neurotransmitter comes and binds to the receptors in the bipolar cells, and the synapse is blocked or inhibited by that neurotransmitter. During the presence of light, the membrane is hyperpolarized, so the neurotransmitter is not released and the synapse is not blocked. During the absence of light, the membrane becomes partly polarized because the cyclic GMP gated sodium channel open, and there is the release of neurotransmitter that gets bind with these receptors in the bipolar cells, and the synapse get blocked. And because of the blockage of the synapse, we see nothing during the absence of light. That's it. Thank you. Teri naina, teri naina, teri naina re. Teri naina, teri naina, teri naina re.